Students, today we study plant embryology. In embryology, we study endosperm. The term endosperm comes from Greek word endos and sperma. Endos means within, sperma means seed. That means someone enclosed into seed. Endosperm is a nutritive tissue formed during the process of double fertilization, where one of the male gamete or sperm fuse with the primary nucleus to form triploid endosperm. The important function of endosperm is to provide nutrition to the developing embryo. Thus, its development begins from starting of embryo development. In gymnosperms, the endosperm is haploid, while in angiosperms, the endosperm is triploid. In normal cases, the endosperm is triploid, but haploid, tetraploid, and polyploid endosperms also reported. However, endosperm formation is suppressed in two angiospermic family, the Orchidaceae and Podostomenanceae. Double fertilization. Its double fertilization means where one sperm or a pollen grain fuses with the egg and the resulting zygote develops into an embryo while the other fuses with the or pollen nuclei or the central cell forming a primary endosperm cell. The primary endosperm cell 3N grows rapidly to become a mass of cell which is known as the endosperm. Now we study types of endosperm. Depending on the mode of development, endosperm is classified into three different types. First one nuclear endosperm, second halobial endosperm, third one is cellular endosperm. According to scientist Davis, out of 288 families, about 161 families show nuclear types of endosperm, 72 families show cellular type of endosperm, and 70 families possess halobial endosperm. First, we study nuclear type of endosperm. Nuclear type of endosperm is the most common type of endosperm in angiosperms. In nuclear type of endosperm, the first and other subsequent division of primary endosperm cell is not followed by cell wall formation. The nuclear endosperm is very common in angiosperm and occurs in around 161 families. The development of nuclear endosperm has been well illustrated in Califa Indica by Johari and Kapil. According to them, the development takes place in the following manner. The first step is the primary endosperm cell undergoes many types of free nuclear division. The cell first begins to enlarge in its size. The enlargement is followed by the nuclear division of primary endosperm. Generally, the endosperm nuclei in the calygeal part of the embryo sac have been observed to be longer than those at the micropylar end. As division progresses, the nuclei are being pushed towards the periphery. So, it's a large central vacuole formed between the endosperm. The central vacuole pushes the cytoplasm containing all the nucleus to the periphery. The cytoplasm thickness and vacuole decrease in size. Finally, a large number of nuclei are pushed in common sheath of cytoplasm and remains at the peripheral side. The multinucleate cytoplasm undergoes cleavage and it gives rise to a multicellular tissue. Johari and Kapil, the first nuclear division are synchronous, later regular, while later nuclear division are asynchronous, irregular types. In case of coconut, the cell wall formation is incomplete. The number of free nuclear division varies in different plants. For example, in case of Coccus nucifera of Palmae and Ericaceae family, primary endosperm nucleus undergoes a number of free nuclear divisions. When the fruit is about 50 mm long, the embryo sac remains filled with a watery fluid or milk containing free nuclei and cytoplasm. When the fruit becomes about 100 mm in length, the liquid shows, in addition to free nuclei, several cells, each enclosing variable number of nuclei. These cells are free nuclei set at the periphery of the cavity, and layers of cellular endosperm are formed. On maturity, it is devoid of free nuclei or cells. In Erica nut and phoenix, the development of this type of endosperm is like that of coconut, but the embryo sac cavity is very small and it completely filled by the growth of the endosperm, and the latter become very hard. In these figures, stage in the development of nuclear type of endosperm. In A, we saw the triploid nucleus. In B, divide. In C, the many free nuclear divisions. In D, we see central vacuole formation. In E, all the nucleus arranged around vacuole on the periphery. Next type is halobial endosperm. It is rare type of endosperm reported in around 17 families of angiosperm, including some monocots. It is the intermediate between the nuclear and cellular type of endosperm. It is the characteristics of the